The sensitivity of DNP-assisted mass NMR spectroscopy allows us to characterize proteins inside intact cells at their endogenous concentration. This application requires careful sample handling.
Cell morphology is preserved and cells are viable when they are cryogenically transferred into the spectrometer using this protocol.
Introduction of the cold rotor can cause moisture and ice buildup, which can mechanically prevent sample spinning up. This demonstration will guide experiments to perform the cryogenic transfer with confidence.
To begin, place a cushion made from a piece of tissue or paper towel under the lid and at the bottom of the cryogenic vial. Place the 3.2 mm sapphire rotor into the cryogenic vial, padded with the tissue paper, with the marked end facing the bottom of the vial. Slow freeze the rotor by placing the vial into a controlled rate cooling container and place the container in a -80 degrees Celsius freezer for a minimum of 3 hours. Transfer the cryogenic vial containing the frozen rotor to a small vacuum flask filled with liquid nitrogen. Use the vacuum flask to carry the sample to the spectrometer facility. Fill a dry, thermally insulated wide-mouth foam vacuum flask with 500 milliliters to 1 liter of liquid nitrogen. Take the cryogenic vial from the transfer flask and hold it just above the surface of the liquid nitrogen to protect it from the atmosphere, while holding the cryogenic vial with